Before the one bonus after credits Marvel question. Erika, do you think there's any way to bridge the gap or answer this question of ancestry using orphan genes?
I think you're going to have a tough time with that, because my understanding is that orphan genes exist at several taxonomic ranks. So there are orphan genes that exist in Homo sapiens only. There are orphan genes that exist in chimps only, in bonobos only. So my answer to that, if you were to use that, would be, okay, well, if you accept that chimps and bonobos are related, or indeed any pair of animals are related that have their own orphan genes, then we're implicitly agreeing to the idea that orphan genes can emerge within kinds. Thus, they can't be used to differentiate kinds.
Okay, do you think orphan genes can be lost over time just through diversification, mutations, speciation events, heterozygosity or homozygosity? Do you think orphan genes that were there can be lost and removed over time?
That's a good question. I mean, I think so. Typically, when we talk about orphan genes, we're talking about the tips of trees. So if an orphan gene had been lost over time, I don't know how we would know unless we directly observed it happening. But I don't know enough about orphan genes. It's possible that there's a way to figure it out.
Because I wonder if today what we're looking at, let's say orphan genes in the great apes and primates in general, that's basically just a subset of the overall orphan genes that were present in all of their respective ancestors. And over time, there's been diversification of those orphan genes. And so that's why they're species specific.
The case that you would have to make for that, I think, would be, you would have to explain why it happens to match the nested hierarchical pattern that evolutionary theory assumes. And I think you would have to also show that this pattern holds and discriminates in other kinds. Because whenever I'm going toe-to-toe with the kinds argument with regard to the great apes, the first thing I do is I always go, okay, how does this map with felids, or how does this map with canids with their orphan genes?
Do you think that the primary explanation, I know there's a few, for the origin of orphan genes is a good, convincing explanation? You know what, actually, before that one, because based on what you said, do you believe that orphan genes fall within a neat hierarchy predicted by common descent?
So it depends on which types of orphan genes you're looking at. I mean, most orphan genes we have are derived from a previous type of gene that is shared by closely related families. So in those cases, yes. Some orphan genes are truly de novo. In those cases, I think that the bigger problem you're going to have is that orphan genes are only called orphan genes because we're looking at them specifically within the context of the tips of these trees. So outside of that context, they're no longer orphan genes. So for example, if humans, Homo sapiens, we have an orphan gene or a set of orphan genes, and then give us 2 million years and 3 other subspecies split off from us, right? Those orphan genes are no longer orphan genes because they're shared by 3 extant species. You see what I'm saying?
Kind of. So like today, if we look at the orphan genes that are present, these taxonomically restricted genes, they, at least from my understanding, they don't follow the pattern. But if you trace them back in time, that's where you get the expected pattern?
No, because orphan genes today, when you say they don't follow the pattern, it's because they exist. My understanding of them is that they exist. They're called orphan genes because they are only in the terminal taxa. So they exist only in chimpanzees, or only in bonobos, or only in domestic cats, or only in, Canis lupus, or only in humans, whatever. But what I'm saying is, if you have, like, let's say a chimpanzee with an orphan gene that bonobos, nothing else has, and it continues to proliferate, it reproduces, give it 2 million years, and 3 subset populations split off from it that are now new species of panins. Right? They're not the same chimpanzee we have today. They've speciated into something new, right? They still all have that orphan gene that was present in their parent population, so it's no longer an orphan gene.
So at that point, okay, so it was an orphan gene at one point. Due to ongoing speciation, it's no longer considered an orphan because you can find it in those lineages.
Right, exactly.
But so eventually you'll land on a spot where you don't find it.
If you could lay them end to end, I would imagine, yes, you could. Like if you had like a full pedigree of 2 million years of chimps, yeah, you could probably find the spot where, if you went back in time where that orphan gene popped up. But then that's kind of an interesting problem, right? Because there was a first chimp where that orphan gene showed up, which means it's not an orphan gene for chimps, right? It exists. It's just a part of the genetic variation of those chimps. It has to move into fixation to now become the orphan gene for the species.
But if we do lose variation and allelic variability over time, and we accumulate mutations every generation, doesn't it make sense that different species are going to be losing different sets of orphan genes at different times? So I feel like the pattern we see, them being species specific, makes sense in light of the nature as to how those are inherited, but also lost.
Well, no. Again, the reason they're called orphan genes is because of their current context. So there have been orphan genes, putatively through evolutionary history that have been lost, but they were only orphan genes when they first showed up in that progenitor species. As they proliferated through their descendant species through allopatric or peripatric speciation, right, they ceased to be orphan genes because they were no longer taxonomically restricted to a single species. The same would be true here.
Okay, so when they first popped up, the typical explanation, obviously gene duplication is one of them, and de novo gene synthesis. Are you personally satisfied with those answers or solutions to the origin of orphan genes, or do you think there's something better out there?
I don't know enough about orphan genes to say, to be perfectly honest. I mean, my understanding is that the current antifreeze protein that exists in cold water cods, not the Notothenioids, but the
Arctic cod one,
but the cold water cod.
Yep.
It's a de novo protein that emerged after a duplication and a series of point mutations to an ancestral protein that wasn't an antifreeze protein. My understanding is that is now an orphan gene because it only exists in those cold water cod. Do you see what I'm saying? So like,
is the cod an interesting example in the sense that we can see beneficial mutations happen, accumulate. We can see the phenotypic effects today in many cases, right? In the Arctic fish example, the Arctic cod, and then also the Notothenioid antifreeze Arctic fish, we're seeing those genes. We understand their effect, their function. In those cases, are we working back and making those inferences that, well, maybe it was a duplication followed by neo-functionalization, and then that novel protein came about with that function? Is there kind of a difference there in how we're interpreting that?
My understanding of how it was done is that these cold water cods have close relatives who are not cold water cods and who do not have that protein or do not have the de novo protein. They do have a sequence of non-functional DNA that matches the cold water antifreeze protein to a T, except for a couple of point mutations, right? So the idea is that if these two are sister species, the ancestor of them both probably looked like the one that wasn't a cold water fish. It has a duplication event, a point mutation, and that's why it's now capable of going into these colder waters.
Okay, so how would you respond to this objection or alternative solution that both species had these proteins or these genes, but one simply lost it over time through mutation accumulation? So now one's present, but rather than that being an evolved protein or gene, it's just that the other species lost theirs.
My understanding of it is that you can tell, I don't know how they do it, but that you can tell which one emerged, right? Like how it's a duplication rather than a deletion.
I wonder how, though, because you're just looking at the two species genomes. You see what's there, but we're working backwards and having to make guesses, inferences, educated guesses, sure, but
I'm fairly certain it's because one of the species that has the mutation in it is like the only cold water cod that has that. And a lot of species that we would agree under both models are like putatively older species, have had far less change to their genomes, have smaller genomes, or, have lost certain characteristics that we would agree are like more primitive, for lack of a better term, that those exist, and that the outgroup is the cold water cods.
It's not possible that the other species also lost their, or maybe this species of cod, they had that gene pre-existing in their system. Now it's been expressed. It enters cold water, and we understand epigenetics, how these genes that are turned off can be turned on rapidly.
So that I don't think could be the case. because this has popped up in separate populations twice, right? So we see it in the Notothenioid fish and we see it in the cold water cods.
Yeah. I'm sorry to interrupt, but if both those species, both those types of fish have those pre-existing genes just waiting to be turned on and expressed, because we understand there's millions of genetic switches in the genomes of biological organisms, really just waiting to be turned on and expressed through environment. And I've got a couple papers that talk about these fish species that have rapidly evolved in real time. They enter a specific environment and then a pre-existing gene turns on. So it's not that that gene evolved, it's just that it's there. And now the environment is making
right, but that's genomically a different thing, right? So adaptation is population level, right? Whereas acclimatization is, which is what you're talking about here, is at the individual level and can be epigenetic. So like these are differentiatable at the genome level. As for how do you know whether or not it emerged in the ice fish or was lost in all of these other closely related fishes? I mean, I think that's just like a pedigree thing. Like you can take the genomes of species and see how they fall, where they fall into these pedigrees by sequencing their genomes. So you would effectively have to argue, much like early folks who were looking at the human chromosome 2 fusion, that in like 10 or 12, I don't know how many different cold water cod species there are, but in every single one, they happen to lose this gene in exactly the same place, and also dozens of other genes in just such a way to make it look like they're the ones that have experienced the change, as opposed to the cold water cod that got it de novo.
Well, it depends how many species there are. Yeah, if we're going to exaggerate and say, well, 1 million species all had to lose that gene, maybe there's only a few, and then there's one that has the gene. Maybe there's a few that have the gene, few that don't. Then that makes sense.
There are like tons of species of Notothenioid fish and cold water cods.
Okay, but would you not believe, so when it comes to parsimony, would you agree that it's much easier to break a gene than build one?
So kind of, but again, I mean, Donny, all of this, as far as I can tell, like listening to you and Bergman talk yesterday, I picked up on the same thing, and I said this in the chat. This boils down ultimately to a difference in how we define fitness and what fitness is. Because
not really with building a gene or knocking a gene out.
Yeah, because it's like if I duplicate a gene, right? I've duplicated that gene. If I add a point mutation onto that gene that gives it a new function, neofunctionalization, as with we saw in the Notothenioid fish, I'm breaking that gene. It's becoming a new, better gene, but I'm still breaking the old gene.
Okay, so you have a gene, pre-existing gene, it duplicates. You get another gene of the same.
Sure.
Neofunctionalization is basically that duplicated gene experiences a set of mutations. Eventually one's beneficial, you get the new function.
Sure.
Okay, so in cases where you don't see that in real time, we just have the genetics. Are they comparing the species and saying, well, here's one gene that's not present, or here's one gene that's only present in one species, but that same gene is present twice in the second species? Is that how they're concluding that it was duplicated?
Yeah, because I mean, it's like, especially in the cold water cods, because it was in the non-functional regions of the genome. So it was like a very specific non-functional section that didn't do anything, that duplicated. Like, and when I say didn't do anything, these cold water cods can swim into cold water, but they freeze to death. So it's not like they swam into the cold water and individually experienced that change. They swim into cold water, their blood freezes, and they die. What happened was one of those regions of the genome duplicates, as tends to happen, as has happened many times in the history of our observing the genomes of organisms. It duplicated and experienced a series of point mutations that neofunctionalized it and gave it the chemical properties of antifreeze. That's what allowed it to go into those new regions.
Wouldn't that gene, the duplicated gene that goes through a series of mutation accumulation, wouldn't that function have to come about rapidly? Because like you said, these fish enter the Arctic water, the cold water, they die. Okay, so how many of them die before that function evolves where they can now live in that water with the protein?
Well, they're not living in that water, right? The way that water temperatures work is they work on a
Okay,
gradient.
gradual. Okay.
Yeah, so it's like their range is in the area just below, as tends to be the case with animals. They push the limit, they live right up to where they can tolerate, and they don't go further than that. And the ones that do die, and they leave nothing behind. So the
So the
ones that
ones that
don't,
gain the ones that gain the function, they're just moving closer and closer to that lower temperature water, this
well,
freezing cold water.
it's as simple as this. If you have cold water cods that live in this area, they can't go into the freezing water, but they're living in water that's decently cold, right? One of them happens to have the mutation, or a handful of them. I mean, I would imagine if they're coming from one mother, one single, clutch of eggs, they lay hundreds of eggs at a time. You're going to get a couple of them at the same time that are going to have this mutation. At least with fish, that's going to be the case. So these guys are fish. They're not plotting their route, but the ones that do go up further north where no other cold water cods can go, access resources no other cold water cods can eat, grow bigger, come back to their home range, reproduce as larger cods, and then pass that gene on to their offspring, which then also have that advantage and that proclivity for going to those more northerly areas.
Okay. So in the fish species of cod that has the purported duplicated gene with the function attached to it, if you want to say it that way, versus the one that doesn't, how do we know that that gene and that function is not just a reflection of the function that is now expressed because of the situation you're talking about? The environment basically dictated what that gene would do in its I don't see how we can rule out the fact that it's front loaded. I feel like a lot of work would have to be done to prove such a thing.
Because the cods that don't have this duplicated gene can't swim into this area. They die. That's how we know.
Okay, so you're saying the ones today that swim into that area, they die. So if it was front loaded, if they were to swim into that area, their gene should be turned on as well.
Right.
Okay,
it would be an example of an epigenetic acclimatization, not adaptation at the population level, which again, to be clear, geneticists can tell the difference between the two. Epigenetics, as you know, is switches turning on and off. De novo genes are entirely new, like structural nucleotide based changes to the genome that are not context specific. They are or they aren't.
It's a fair point. So would you say that a lot of these genes that are just waiting to be turned on through environment, they're not really active at the moment, right? Until that environment triggers it to be turned on. Would you consider those redundant then in many ways?
Which genes?
Well, if you have a gene that would turn on in an environment, let's say hypothetically you had a fish that entered a certain environment, and in this case, it was an epigenetic change.
Can epigenetics do things that drastic?
Well, I think if the gene's just there waiting to be turned on, waiting for the right environment to allow it to basically pop up and perform a function.
Right, but we would see that when we sequenced it. We would see that it was a gene that was there but turned off, right? We would see it in the genome.
So what if the gene, while it's turned off, it's not active, it's not expressed?
Sure.
If it's redundant and not essential at that time, let's say in these codfish, these Arctic fish, maybe mutations destroyed those, because obviously those redundant elements would not be under high selective pressures, from my understanding, because they're not essential. So some species get theirs knocked out through mutations, while others,
it could drift in and out. Like, let's say the climate changes and the area where these codfishes live suddenly becomes warm and they don't need this antifreeze protein anymore. Is it possible it's going to go away? Maybe. I mean, I don't, unless it's causing their cells like strain to produce, I don't see why it would, but it might just because of drift. But again, the critical point here is that the gene doesn't exist in other cold water cods, right? It's not something that exists in them and is currently turned off. It exists in the cold water cod and is always on. It's not something that is regulated. They always have antifreeze in there.
Well, because now they're in that water permanently. That's their habitat. That's their environment. So it's
they're always there. Now, here's what you could do. Take one and put it not in cold water. I don't know if anybody's done that.
Yeah, see if it
turns off. I don't think it will, because a lot of animals that are like...
Well, we can make that could be a prediction right there. That's testable. Will it turn will it turn off or nothing happens? Maybe it was truly due to duplication
I
followed by neofunctionalization. So
I
in the case you were talking about, those other species, yeah, they could have had those redundant elements or genes wiped out through mutations, while the other species actually did enter the environment in the way that allowed for the gene to be turned on.
I mean, that's so that second part is going to violate parsimony, like how parsimony works typically with these kinds of things. Which, if you're gonna, you can violate parsimony, but you have to have a really good case for it, phylogenetically speaking. Um, Especially because, again, I would imagine we would agree that all of these cold water cods are related. But then, secondarily speaking,
I'd
I'm
have to see
speaking.
how many, but probably, yeah. But I mean, there could be multiple kinds of
Sure. I
codfish.
mean, that's for y'all to
Okay,
figure
to
out.
clarify, Erika and Samir, I'm sorry. I could talk to you about this all day. It's an interesting topic, Erika. So here's the last question I'll ask out of curiosity. So
No, just before you ask, Donny, I just want to say that you can make your case better with the African lungfish that buries itself in the dirt for up to a year or more.
well, it's just because, I mean, I could pull up the papers I'm talking about with rapid evolution in fish. It's just because I'm directly engaging Erika's Arctic cod example. Is this truly beneficial mutation, duplication, followed by neofunctionalization? And so, and I know there's been a lot written on it. And so, Erika, you believe it's more parsimonious to say that these genes or these antifreeze proteins were built up through
not
duplications and
not only do I
mutations?
think not only do I think it's more parsimonious, I bet that there is a way that they have distinguished this and precluded epigenetics as an idea. I just don't know what it is. I bet they've
Okay.
done that.
Okay.
And it's something that, if I had the time, maybe sometime in the next couple of weeks I can look into. I don't intend on it, frankly. I've got a lot of stuff on my back burner right now, but I mean, it's worth looking into. If you think you can make the case, go for it. Again, eventually I'm going to get around to it, and I'll see what I see. But I don't think that that's a case that can be made. And if you want to double check that, you should ask Jackson Wee, because I think he has worked with some of those fish before. He works at an
Yeah,
aquarium.
he and I discussed that 5 years ago or so. Okay, well, no, that we got Y chromosomes to work on. So yeah, we'll get to that eventually. But I think these are some good thoughts and considerations on the topic and the issue, because I'd really like to map out how many of these beneficial mutations are just truly beneficial with very little reductive effects versus the ones that are okay, beneficial, context specific, like you say, Erika, accurately, but at the genotype, reductive and functionally compromising in many ways. Because the Notothenioid one and the Arctic cod one, if not due to epigenetics or not due to front-loaded DNA, I don't really see any reductive effects from it. You know what I mean? So that'd be an
Right.
interesting one to
I
see.
mean, it's right, because it's a de novo protein that, for all intents and purposes, it's like I teach my students. There aren't very many, but there are mutations out there that I struggle to see, a situation where it would be negative for the animal that has it. Likewise, there are situations where I struggle to see benefits to many different kinds of mutations, even though I would say that the vast majority fall into that context specific framework. Uh,